There we go. All right. So you guys can hear me okay on Zoom? Just want to check before. Oh, everyone's like,
Ask them to give like thumbs up.
okay, cool. So today we're going to be covering the cell cycle, but the second part of the cell cycle of the first part. So we'll be covering like G1 basically. Last review session was covering the overview of the cell cycle and a couple other mechanisms, but now we're going to be talking about the pre-RC and IC formation as well as activation of CDK. So first we'll start off with mechanisms of control and regulation in G1. Here's like a little overview of G1. Before the cells enter the cell cycle, they have to like receive signals for growth. They have to check for DNA damage. They have to check for cyclin, and then they also have to make sure they have all the energy, nutrients, and size requirements to continue on. They also have pre-replication complexes formed, which will go more in detail now.
Alrighty. So the early G1 formation of pre-RC, it's formed in early G1. Am I in the right slide?
Mm-hmm.
Yeah. So the pre-RC stands for pre-replication complex. And it's basically a protein complex at the origins of replication, and the cell has a lot of origins of replications. I think Laura gave like an example that eukaryote or us humans have like 300 or actually more. I remember somewhat like that. And we here at the beginning we have CDK activation. Actually, we have it. We have the complex of pre-RC formed at low levels of CDK activation. So that is at the beginning of G1 phase, before the restriction point. So before we have CDK activated and like moving on into replication. And the reasons that we begin the pre-RC complex at the beginning of G1 is because CDK inhibits the formation of pre-RC, and we're going to talk about it more next week because it inhibits re-replication while the cell cycle is going on. It's also important to know like that organisms have specific areas for DNA where the replication starts, and this is like the same thing as the origin of replication.
And so for the formation of pre-RC, the start of the formation begins with the ORC DNA binding protein binding to the origin of replication. And keep in mind, like there's many origin replications along each chromosome to help start off the process of DNA replication, which we'll talk about in the next week's topic. And so after binding as an ATPase, it'll recruit, or ORC will recruit CDC6, also an ATPase. And then this will recruit CDT1 and then MCM27, which is also known as genomic helicase, also an ATPase. And that MCM, which, like I just said, is helicase, will then be used later on, as you'll learn, to unwind the DNA. So once this whole MCM complex is loaded, CDT1 will dissociate, and the completion of this process is called origin licensing. And so the origin of replication in that area is now set up for the rest of the rest of the process to continue. And the next step for this particular complex will be to become a pre-IC, and that will form after the restriction point, but that requires active CDK. Sorry, one last thing I forgot to mention here is that you have to make sure that there might get a question too about like, is there going to be pre-RC formation in early G1 if you have high levels of CDK cyclin activity, And the answer is no.
Okay, so as Gabe said, that for the pre-IC complex, which is just a bigger protein complex, we need CDK to be activated. And last week, or at the beginning of the week, we talked about V1 and CAK. And we said that once the CDK cycling complex is bound to a phosphate from CAK, then it's activated. But if it's at the same time bound to V1, then it's going to be inhibited, even though if it has the activator phosphate. And so in order for us to activate the CDK cyclin, we will have to phosphorylate it or dephosphorylate the inhibitory phosphate from V1. And do you want me to talk about this slide or?
I can do that one.
Okay.
Oh.
Are you gonna talk about this?
Yeah, I can do both. I don't mind. Yeah, I don't mind. Okay. So now that you have CDKs activated, it's time to activate the rest of the pathway in order to get the pre-IC formation going. So in order to do all this, what happens is you'll have a growth factor come in. You'll have the RAS pathway activated. As you guys have learned, that whole pathway ends up activating CDK cyclin as its like end result. And then CDK cyclin will then go ahead and phosphorylate the RB protein, right, as you can see right here. And then once it phosphorylates the RB protein, that causes RB to let go of E2F and allows it to, I just want to show this picture here. It allows it to transcribe these genes that are needed for S phase. And then these genes transcribe, or they code for proteins that once transcribed and translated, these proteins will help initiate S phase, but also a lot of them will be involved in the formation of the pre-IC complex. And so the pre-IC complex, once it's formed, allows the recruitment of DNA polymerase. And then that's when, after that, origin firing can occur, which is the start of S phase. And so this whole process right here, well, okay, first I should probably introduce this right here. So a good way to remember like what is going on here, right? Because it's not like CDK is like directly, it's activating, active CDK cyclin is activating E2F, but it's not doing it directly. The way it does it is it basically by phosphorylating, a good way to think about it is like RB is holding E2F hostage. So cyclin CDK needs to phosphorylate RB, which means basically taking it out from being able to do its job. So once RB is like deactivated, disarmed, you could say, E2F is free to go and do its job, which in this case happens to be translate or being able to just transcribe these genes that are needed for S phase. So yeah, it's like cyclin CDK is when active is indirectly activating E2F. And a lot of this, like what we talked about this week, it's kind of hard to visualize like how it's all put together. So I've drawn a little diagram. Please excuse my terrible handwriting that looks like a small child drew it. But basically, here you can see the start of the cycle of like G1. So the time goes this way. Probably should have drawn that in. But basically, early G1. In early G1, you have like no, at the very beginning, you have like no pre-RCs, right? So this is a DNA strand right here. That's a chromosome. And along the chromosome, you'll have at these like origin replications that'll form, you'll have pre-RCs begin to form. But that's only, this formation's only going to happen, like I said, if CDK cyclin is like present, but only in like low levels of like activation. So like if it's in high levels, it'll prevent that from occurring, because as you'll learn like later, you don't want that to reform. So like, so your DNA only replicates like once. So when you have low levels over here in early G1, the pre-RC will start to form, but then those pre-RCs, after crossing the restriction point and then going, once they cross the restriction point, they're in late G1. And again, restriction point is the point of no return. Once they're in late G1, that's when pre-IC formation will occur. And then the restriction point, of course, is the point at which CDK begins to be activated, as shown in the process in the last slide, with this whole RBE2F hostage situation going on. And so once CDK does its job, as shown on the previous slide, that's when this pre-IC formation can occur. And that's what you have right here. These are now called pre-ICs. As you can see from these extra dots, there's extra proteins recruited onto those complexes. And yeah, like I said, those are called pre-IC now. And then afterwards, down here is when you'll have origin firing. Just didn't fit on the image, but you'll have origin firing, which will then be, if you were to continue this way in time, it would be S phase. So now we have some questions for you guys.
Okay. So the first question, is CDK active throughout G1? Yes. Is it just only at the beginning of G1, or is it only before the restriction point, or is it not active at all in G1?
If also feel free to answer like on Zoom or whatever, like in the chat. Whoever like gets all like the most correct, like you will get 15 extra TA bonus points, which counts towards the TAs liking you the most. So these are really high stakes here, guys. Please try your hardest. If you get it wrong, it's okay. It's okay. You still get 5 points for effort. So might as well.
I think B.
B.
Okay, why?
Because if it was active all throughout G1, then it would be doing things too early, like activating E2F too early, causing creation of the pre-IC too early. So it's only going to be active before the restriction point, and you want those things to happen.
Yeah, so it would cause like activation. You're right, it's B. And the thing is, we don't want it to start at like the beginning of G1, because we said that CDK activation causes inhibition of the formation of pre-RC, and we're gonna talk about like how it prevents it and like why, which is like to prevent re-replication of the DNA while we're already like in one cycle, because that's not good for the cell to have like double chromosomes. So yeah, we only have it only before the restriction point to for the activation of the pre-IC complex, which Gabe was just talking about.
Yeah.
To get activation of the E2F transcription gene.
Yeah.
Good job.
It would be after.
Yeah.
Okay. But it says before.
Oh, before. Yeah, it's before. Sorry. I thought you were asking. if
No,
I
I'm
can do
saying
the
like
question.
active after. All right, anyways.
So
let's
wait,
move on.
isn't it active like at the restriction point before to activate the E2F transcription gene?
Yeah, well, it's like that is the restriction point. Anyways, we'll move on.
At the restriction. I don't know, that's what her note says.
Yeah.
Okay, so for question 2, which of the following are examples of ways in which a cell regulate the cell cycle? And there could be more than one choice.
Also, you can. Oh, it's even worse. All right, guys, get your
Anyone has
answers
a
in.
question? An answer.
No
one has an answer.
Unfortunately, the chat on Zoom is a little inactive, but
I mean, Luke answered the first one.
oh, okay, never mind. My bad. Thank you, Luke.
And I think it's A, not B, not C, right? But in TDK, negative, probably.
Think about it. We talked about it a little bit on the question before that one.
The cell regulates the cell cycle.
So you guys said A is right. Does anyone want to say why? Nice, Luke, you're right.
Okay.
Well, because, well, it needs to be phosphorylated by CAT, CDC, and it's also phosphorylated by V1, but CDC 25 is get rid of that phosphate in order to activate it.
Exactly. It gets rid of the phosphate that is but by V1. That's right. The
Oh.
Luke, you said it. D is also correct. Do you want to say why in the chat or unmute yourself and say why?
Oh.
Did you get that?
Yeah, I did.
Oh, sorry.
Very good.
My audio wasn't working for a second. It's because like it can phosphorylate things, but this can also have a negative consequence if it's not properly like checked, and so it can cause like a damaged cell to go through. Right?
Yeah.
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 So
So
that would be the negative, and then the positive would just be normal cell function.
exactly.
Yeah.
Yeah. So D could we said that kind of like the negative is that it inhibits the formation of pre-RC if it's active, which inhibits a re-replication of DNA. And as you guys said, the A is also correct. Just for what's your name? Nick. For what Nick says that it gets phosphorylated by both V1 and GAC, and it also gets dephosphorylated by CDC 25, which takes off the inhibitor phosphate. B is not correct because it here it says that RBA, it causes like RBA to bind to ET of I can't say E2F transcription factor, but it actually causes RB to let go of E2F. And C is also not correct because pre-RCs form at the beginning of G1 phase, not during S phase. And that's all we had today. So if you guys have any questions
Or
feel
any
free to
applause.
ask, or any applause.
Feel free. Does anyone on Zoom have any questions about the problem set? Wait, or have any questions in general first, and then
Yeah,
we'll go over
don't get
problem set
started.
questions. To do. We're gonna stop the recording now. Yeah.